Time no see. We are going to be taking some notes today. Hope you guys are doing well. We are going to do a 3-step process here. So, first thing I want you to do is to go and make sure you have this either printed out or have a blank sheet of notebook paper that you can fill this in as we go. We're going to be taking notes just the same as we would be doing if we were normally in class. So, I'm going to be going through this slideshow. You're going to be filling in the blanks. If you can't print it out, just write it down on a sheet of paper. So then that way you have it for reference, because at the end, we are going to be doing this check-in. That'll be the last step. It'll also be posted on Haiku along with everything else. Just keep scrolling down under e-learning assignment number 2. And later on down the line, we will even be having a little quiz on mitosis and meiosis. So please make sure you are taking good notes down on this note sheet, and let's start. All right, so this is all going to be about cell division. It's going to fall into 2 categories. Those categories are mitosis and meiosis. Mitosis is when you have one cell that becomes 2 identical cells, and that's going to be a body cell. So that's going to be like your heart cells or your muscle cells or your skin cells. They all undergo mitosis. Meiosis is when you have one parent cell that divides twice to make 4 haploid cells, meaning they have half the amount of DNA. And that's going to be sex cells. So that if you're a boy, that's going to be sperm. And if you're a girl, that's going to be eggs. Those previous slides right here didn't really matter. So this is the first one you're going to start taking some notes on. Cell division, as I said, is a process by which a cell divides into 2 new cells. Why do cells need to divide? Living things grow by producing more cells, not because each cell increases in size. You want to divide to repair damaged tissue, and if a cell gets too big, it cannot get enough nutrients into the cell and waste out of the cell. One thing I wanted to point out to you is, unlike in class, I am not really going to pause to give you time to write, because this is a video. So you can just push pause on your own and write, and then resume when you think you're ready to go. It's the wonders of technology. All right, so key things to know: cells divide to grow the organism, to repair damaged tissue, and so that it doesn't get too big and you can't get waste out of the cell. All right, cell division. The original cell is called a parent cell. So here we have one parent cell. This is our original cell. 2 new cells are called daughter cells. Daughter cells are identical to each other and to the parent cell. So this is for mitosis. So here's the parent cell. This is the same parent cell that's right here. But what happened was this part. The DNA replicated. See how there's like one line each here, and over here it's paired up? And there's 2 lines and they form like an X? That's because all the DNA replicates, so it all has a copy. So before cell division, the cell replicates all of its DNA so that each daughter cell gets a complete set of genetic information from the parent cell. So parent cell, DNA replicates, more lines, and now we have 2 daughter cells coming off right here that are genetically identical to this cell. So we just finished learning about DNA not that long ago. That means that this cell and this cell have the exact same DNA that the parent cell had. Nothing different at all. Same bases in the exact same sequence. So, when we talk about DNA, we are often talking about chromosomes. Body cells are diploid, which means they have a full set of chromosomes. So, for humans, a full set of chromosomes would be 46 chromosomes. And here they are all laid out. So, there are 23 pairs of chromosomes. So, here's They're all numbered down to 22. So 22 pairs. 1, 2, that's a pair. 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 And then at the end, you have your sex chromosomes. That's the 23rd chromosome. This is a boy, and I know it's a boy because it has an X chromosome and a Y chromosome. Easy way to remember that. If you got a Y, you're a guy. If you don't have a Y chromosome, you're a girl. So a girl would be X and X. All right. Sex cells are haploid, meaning they have half the set of chromosomes. So I'll often say haploid like haploid. It has half the amount of chromosomes that you're expecting. So a human sperm will have 23 chromosomes. A human egg will have 23 chromosomes. Getting back to this and how each one is a pair, you might have heard of the genetic company 23andMe, where you can get DNA testing to see where your ancestry is from. They're talking about the 23 pairs of chromosomes and me. So those 23 make up you. All right. Cell cycle. These are the series of events that cells go through as they grow and divide. There are a few of them here. We got G1, which is growth phase, S, which stands for synthesis, G2, which is another growth phase. Then we have the mitotic phases of prophase, metaphase, anaphase, telophase. This is when the cell is getting ready to divide, and then cytokinesis, when you actually divide, division of the cell membrane. So think of it like the life cycle of a cell. You start out from your parent cell, and then you get quote unquote born. Your parent cell is replicated. Here you are along with the other daughter cell. Then you go through a growth phase. Then you replicate your DNA. You grow again. You get ready to divide and make daughter cells from yourself. And then you divide. That's what it's all about. These three here, G1, S, and G2, make up this whole thing, which we call interphase, which is meaning the phase in between cell division. All right. So you're created from your parent, you grow, this is the interphase, then you get ready to divide yourself, and then you make more cells. All right. So mitosis is the division of the nucleus. Prophase means the preparation phase. That's when the DNA coils and condenses into chromosomes. The nuclear membrane disappears and mitotic spindles form. Now, one thing I want to point out to you is if you see down at the bottom of your screen over here, you don't need to memorize these steps. All right. So these steps, prophase, I'm about to talk prophase, metaphase, anaphase, telophase. You don't have to have them memorized. I'm just letting you know so you know how it works. So here's your chromosomes, these big X's here. This nuclear membrane, the nucleus, is going to start dissolving, and then you're having these mitotic spindles forming. This is what it looks like in real life. Here's a cell that's in interphase. Notice you can't really see the chromosomes. They are still there, but you can't see them because they're not condensing up because you're not getting ready to move. If you've ever moved in your life, you don't go from just having a room that is laid out the normal way that you have it to automatically a new room. What do you have to do? You have to put everything in boxes. You have to condense it up. And that is what's happening here. Here's a room all spread out. Here's a room that's getting ready to move. They're putting everything in boxes. All the chromosomes are condensing together, and then it's going to split and form two new cells later on. The next step of mitosis is going to be metaphase, which is the middle phase. Chromosomes are going to line up in the middle, which is what you're seeing right here. And then the spindle fibers attached to the centromere. So these are the spindle fibers, these long lines here. And they're attached to the centromere, which is kind of like this middle part. Center, like centromere. Attaching right here. And what's going to happen in the next phase is they're going to start to pull, and these chromosomes are going to split. Let's see if we can see that happening in just a second. Here's the real-life image of it. Chromosomes in metaphase, you're seeing that it's still one cell, but they are lining up in the middle. They're getting ready to split. Anaphase, which I know sounds lame, but it's my favorite phase of mitosis. It's when things actually start moving. You see that these start to get shot off to one direction. They used to be in X's here in the middle. Now they're splitting, and they're no longer an X. They're kind of just like half an X on each side. So, chromosome split. Sister chromatids are pulled apart. It was a chromosome when it was in the middle. Now that they've split, they're each called sister chromatids. So this is a sister chromatid. This is a sister chromatid. Sister chromatid, sister chromatid, sister chromatid, sister chromatid, and so on. Spindle fibers move to the opposite end, so they're pulling them down over here. Here's what it looks like. You're seeing those chromosomes moving away from each other, or rather, I should say sister chromatids. Telophase is the ending phase of mitosis. Chromosomes begin to uncoil, so you're almost at the point where you have two cells. You don't quite have it yet because they're still connected right here, but here are the sister chromatids that are separated from each other. There's one orange in each one purple in each one red in each one blue in each one, so that this way it's the same genetic information. The nuclear envelope reforms. You can see it coming around here. The nucleus is coming back. The spindle disappears, so we no longer have those lines that are connecting everything, and cytokinesis begins. Cytokinesis means the movement of the cytoplasm. So cytokinesis is when this thing gets split and you form two fully separate new cells that are genetically identical. The whole point of mitosis is you're making two genetically identical cells. All right, here's what telophase looks like. You can see they're still connected right here. Then we got cytokinesis, division of the cell membrane. Like I said, it's like you took like a cleaver or a sword and you chopped them in half right there. And now you have two new cells. The chromosomes begin to unwrap and you get to spread out in your room, just like you would if you got done moving. Okay? Now, cancer should be something that is pretty fairly important to you because a lot of people might deal with cancer at some point in their life. What cancer is the uncontrolled process of mitosis. It's rapid cell division. So, DNA controls all cell division. Some cells lose their ability to control the rate of cell division. The DNA becomes damaged or changed or mutated. These superdividing cells form masses called tumors. So here you have a bunch of normal cells. Here are cells that are dividing too rapidly. It's like they're rebelling. They are not following the rules and dividing when they're supposed to. They're dividing constantly all the time. And by doing this, they form this tumor, and that's what we call cancer. And it causes other problems throughout your body because you don't want cells growing and tissue growing where you don't want it to be growing. That causes body problems. There are two types of tumors. You got benign tumors, which are not cancerous, and these cells do not spread to other parts of the body. All right. And malignant tumors, which are cancerous, and these cells break loose and can invade and destroy healthy tissue. So here you got a benign tumor, which is contained within this membrane. Malignant is when the rapidly dividing cells start spreading throughout your body. So in terms of cancer, it often becomes a greater problem if it becomes malignant, because rather than the problem being contained to one area, like let's say you have lung cancer, if it's malignant, these cancerous cells are spreading to other parts of your body through your bloodstream. So it might start off as lung cancer, but if it gets malignant, it could spread to, like your heart or intestines or something like that, and cause more problems over there. Some terms you might want to know about cancer. Carcinomas result from the altered epithelial cells, which cover the surface of our skin and internal organs. Most cancers are carcinomas. So most cancers are basically on the skin, whether that be the skin that you see on, when you look down at your body, or the quote-unquote skin, not really skin, but the protective layer that is around your internal organs. Sarcomas result from changes in muscle, bone, fat, or connective tissues. Leukemia results from malignant white blood cells, meaning blood cells which attack your own body. Lymphoma is the cancer of the lymphatic system, cells that derive from bone marrow. Melanomas are cancers of specialized white blood cells that make antibodies. Meiosis. So this is the other side of the coin. Mitosis Again, I know I'm repeating myself a lot, but I'm really just trying to hammer it into your head because it's an important point, and it's one you're definitely going to be tested on. Mitosis is when you take one cell, you divide it into two identical cells. For meiosis, you're going to take one cell and you're going to divide it into, eventually, 4 genetically different cells. And they're always sex cells. Meiosis is always body cells. So, the meiosis is the process of creating sperm and egg cells. It's called either spermatogenesis if you're a boy or oogenesis if you're a girl. Haploid cells is what is produced through meiosis. Haploid like haploid. It's going to have 23 chromosomes because normally we would have 46. So if you were to take a sample of your skin tissue, it would have 46 chromosomes in it because that's a body cell. But if it's a sex cell, like a sperm or an egg, it's going to be 23. 2 divisions reduces the number of chromosomes to half. If there was a mistake when chromosomes are separating, then the resulting sperm or egg will have too many or too few chromosomes. All right, here's what it looks like. It's a little bit more complicated than mitosis, which is why we usually start with mitosis. But in the end, it's a very similar sort of process. So you're going to start off with one cell. The DNA is going to replicate. Remember how before it was one chromosome? It's going to replicate here, and they're going to have these X's. So here's this guy. It's going to replicate, make its own X. This guy, it's going to replicate, make its own X, make its own X, make its own X. Then it's going to split. And you're going to notice that it's not always the same color. And we're going to talk about why that is in a second. And then it's going to split again. And you're going to have these 4 genetically different cells. And you can tell they're genetically different by the differences in coloration. All right. So these are the processes that a cell is going to go through. If you want to pause and read it, feel free. It's basically the same processes that would be in mitosis to start out with. So you got it, you duplicate, and you have something over here that's called crossing over, actually here, which I'm going to talk about in a second, and then it splits. How many chromosomes does each cell have? It's going to be 46. Then you have these, it's the same as what was on this side after it split. It's just continuing on. I couldn't fit it all in one slide. It's going to follow this path, I should say, and then make 4 genetically different daughter cells. And you can tell that the genetics are different by the colors here. They're all different. And at the end, how many chromosomes each cell have? It's going to be 23 different chromosomes within. I'm only showing 2 just because it'd be very busy if I put in 23, but it's 23. All right. So how can siblings look alike but not be exactly the same? How does that make any sense? Here we have Venus and Serena Williams. They are sisters, but they don't look exactly the same, even though they had the same parents, the same mom and dad. And if you have brothers and sisters, you probably don't look exactly the same as they do, even though you had the same parents. This is because of 2 things that happen in meiosis. So the first thing is crossing over. For crossing over, the chromosomes during prophase 1 undergo crossing over, where parts of the homologs randomly switch places. Homologs means same. Remember, homo means same. So if you follow my mouse cursor here, this is one chromosome. This is another one. Here's one chromosome. Here's another. All right. And they are going to bond to each other and randomly switch places. If you follow the arrows over, you see how they kind of like overlap here? And their legs are going to trade. So you used to have the pink chromosome where all the pink is just on the pink. And then all of a sudden, some of the pink is going to come over here. And it's going to trade places with the blue. And now the blue's over here. It's this swapping of genetic information. Now, this is important because if you think back to you and how you're made, you got DNA from your mom and DNA from your dad. So let's think of this blue chromosome as being the chromosome that you got from your dad. And this pink chromosome is being the chromosome that you got from your mom. They are genetically different, your mom and your dad. So, during crossing over, you make new chromosomes that have, like this one here, has most of your dad's DNA, but a little bit of your mom's. And this one over here has most of your mom's DNA, but a little bit of your dad's. And this is just at the start of meiosis, and things are going to get mixed up even more. And that's how you get these different genetic combinations. And that is why, one of the reasons why, you would look different than your brother or your sister, and why everyone has different DNA except for twins, which I'll get into later. All right, so importance of crossing over. Gene combinations that a person gets from his or her parents will be different to varying degrees than the combination of a sibling than the sibling may get, which is why, you look different. Independent assortment is the random assortment of parental and maternal chromosomes. Sexual reproduction creates a unique combination of genes. Independent assortment of chromosomes is in meiosis, and it's makes for the random fertilization of gametes. All right, so talking about independent assortment, like I said, you're seeing that crossing over happening right here. And the red is going to, this little leg of the red is going to go on the yellow, and that's what you see over here. And this little leg of the yellow is going to go on the red, and that's what you see here. Independent assortment means how things line up during the process of metaphase, all right? So this is probably the most complicated one for students to understand, but this yellow, mostly yellow chromosome could have been over here, or this mostly red one could have been over here. What side they wind up on is just random. And it's that randomness that contributes to the genetic diversity, because as you go down the line, let's focus on this yellow one here, mostly yellow. It's going to get pulled to the side, and now the yellow one is here. And if I continue on, here's the yellow one, and it splits. And again, what orientation it has is independent assortment, so it could have been that the red leg part was on this side and it splits. And then it winds up over here. So the yellow one in this last, it's called gamete. It's the last cell that's made. The yellow one is with the blue one. But it didn't have to be, because way back over here at this step, if the red one was over here, the red one would be on the same side as the blue guy. And then it would be, now it's the red one that's here, and the red one that's here. And it shouldn't be yellow, but now it's red. Now it's red. Now it's red. And it wouldn't be yellow here. Now it's red and blue. So how it lined up way back over here, whether the yellow one's here or the yellow one's over here, determines in the eggs and sperm at the end what sort of genetic combination you're going to get. Okay? All right, let's go back to where we were. So key things to remember: meiosis, you're making 4 haploid cells, so it has half the amount of DNA. You do this because if you have 23 chromosomes for your mom and 23 chromosomes from your dad, how many chromosomes are you going to get? 46 chromosomes. And hopefully that makes a little bit of sense to you, because what that means is it's a normal body cell. So from this start, when you were fertilized and the sperm and egg combined to make you had the correct amount of chromosomes. When people don't have the correct amount of chromosomes, that's when issues start to happen. So if you've ever seen or met or saw someone who has Down syndrome, they have 3 21st chromosomes. And this causes some issues. Oftentimes, if it's the incorrect amount of chromosomes, so let's say that it's a 22 and 23, that something happened to either the mom or the dad with the sperm and the egg, but the baby just will not be born. It would be a miscarriage. So by having 23 chromosomes and 23, you get to 46, then you have, hopefully, a normal, healthy baby. All right, back to this. So, karyotyping. Karyotype is a photomicrograph of chromosomes arranged according to a standard classification. So, like I said, 23 chromosomes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23 chromosomes once you count this. What is this, guys? Boy or a girl? That's right. If you got a Y, it's a guy. All right, so this is a boy. 23 pairs of chromosomes. Chromosomes are digitally arranged, so they are matched with their homologue or partner chromosome. So here's one chromosome, here's its partner chromosome. It's its homologue, all right? So all these are homologues. Homologues here, homologues here, homologues here. Homologue chromosomes are the same size, shape, and carry the same genes, and one is inherited from each parent. They are numbered according to size. So you start off with the really big ones, and then as you notice, as you go on, it gets smaller. As I said before, your 23rd pair is going to be sex chromosomes. If it's XY, it's a guy. If it is XX, it's going to be a female. For you guys out there, myself included, the Y chromosome is kind of a dud. It doesn't really carry any genetic information. So that's why, as we'll learn about in about a month or so, females, are more disease resistant because they have two good copies of the X gene. The Y chromosome doesn't really have much going for it, so we only get one good copy. So we're more prone to things like color blindness and hemophilia, which is where you bleed very easily, because we only have one good copy of the X chromosome. With everything else, we got two copies. So let's say that this chromosome, there's some sort of genetic problem with it. Well, we got a good copy, and so we can still make proteins off of this guy, and we'll still be perfectly fine. So that's why everything comes in a pair. Gives us a backup. All right. So, abnormal karyotype. Oh my God, can't talk. Abnormal karyotypes, which means alterations in chromosome numbers. Non-disjunction occurs when either homologues fail to separate during anaphase of meiosis 1, and this results in some chromosomes having more than one copy and others only having one. So trisomy is what I talked to you about before. That's people who have Down syndrome have trisomy 21, which means they have 3 21st chromosomes, which means that way back over at, what step would it be? It would be this step right here. Rather than these two splitting, this whole thing would just go to one side. And so you'd wind up with both this yellow and red and this double yellow in here. So it'd be blue, full yellow, yellow and red, all in the same chromosome. And when that would be combined with the sperm or egg, you would have too much DNA. All right, because you'd have 3 21 chromosomes by that happen. Monosomic means a cell that's missing. Is a cell that's missing one chromosome. So that's Turner syndrome. Here's trisomy 21. Like I said, 3 21st level chromosomes. It's a guy. Klinefelter syndrome. You have 2 X chromosomes and a Y. So people like this generally look like a guy, but have some feminine characteristics as well. But same rule still applies. If you are a Y, you're a guy. Unfortunately, people who have Klinefelter syndrome are usually sterile because of the double X. All right, so other chromosomal disorders can be caused by these things. So deletion is when you get rid of something. So for all these, I'm starting on the left, going to the right. So over here it goes A, B, C, D, E, F. The D is going to get deleted, and it's going to go A, B, C, E, F. All right, so you're deleting part of your chromosome. That might cause a disorder. Inversion is when it gets flipped. So the D and the E are going to trade places, and it's going to be A, B, C, E, D, F. So when you are trying to make your protein, it's going to be in reverse, and you're not going to make a good protein, and that might cause a disorder. Translocation is when part part of it moves and you aren't expecting it to. Usually this is problems with body cells, because you kind of almost want this with meiosis because it's leading to genetic diversity, but you don't want this with body cells because you want to keep things the same. So you don't want part of your gene ending up on a different chromosome where it won't be read. And duplication is when you take something and you duplicate it. So over here there's 2 Ds, A, B, C, D, D, E, F, and that would result in potentially a bad protein as well. All right, so that is it. What I'd like you to do now, make sure your notes are all done, and then go on over and try out this check-in when you're ready. Fill it out, submit it, and we'll see how you do. Thanks.